Welcome back to the Deep Dive. Today we are tackling, well, quite a stack of sources, really getting into the weeds at this highly specialized intersection of immunology, vaccine development, and the sometimes frightening complexity of human genetics.
We're looking right at the frontier of vaccine safety, specifically this persistent challenge, ensuring protection without accidentally triggering autoimmunity.
Right, and our listeners shared some really technical material here, focusing on how we use animal models, specifically rodents and non-human primates, and what their limits are.
Exactly. It's a deep dive into the limits of prediction, you could say.
Right.
We're analyzing their dual role, which is foundational, obviously, for validating vaccine efficacy and basic safety.
Foundational, yes. But the bigger question, the critical one we're unpacking today, is about their inherent limitations in predicting a very specific, very human risk, a thing called molecular mimicry.
Molecular mimicry. That's the potential short circuit, isn't it? Where the immune system gets confused and starts attacking self because it looks like the non-self invader, or in this case, the vaccine component.
It sounds simple when you say it like that, but the implications are huge. Vaccines are, of course, essential, but when the goal is immunity and the risk, however small, is autoimmunity, well, you have to scrutinize that gap.
The immunogenetic gap, it's massive between lab animals and the incredibly diverse human population. But at the same time, we need to drill down into why they become, well, genetically blind when the risk is driven by these highly personalized human HLA presentation profiles. That's the crux of it.
Okay, let's start where a lot of the crucial early testing happens. Non-human primates. Rhesus macaques were, really high profile during the SARS-CoV-2 vaccine development, like for BNT162b2. Why are NHPs so indispensable there? What are they telling us?
Well, NHPs give us what you might call a high-fidelity snapshot of the protective immune response. They're physiologically quite similar to us, so they offer strong predictive power about the type of immunity we can expect.
Okay, so not just does it work, but how does it work?
Exactly. With the BNT162b2 vaccine studies, for instance, macaques were absolutely essential for validating efficacy, sure, but maybe more critically for confirming the precise T-cell polarization.
T-cell polarization, that directionality of the immune response, that's key for respiratory viruses, isn't it?
Yeah.
What profile did they actually confirm?
They confirmed a distinctly helper T-cell type 1 or Th1 biased response. And that's really what you want for a respiratory virus vaccine. Historically, a Th2 biased response in some older models, not necessarily these ones, but historically it was linked to potentially unwanted outcomes.
Like vaccine-enhanced respiratory disease, that kind of thing.
Potentially, yes, or just not the optimal kind of protection. So the NHP models were used to definitively show the response was leaning heavily towards Th1, which is generally considered protective and favorable for viruses like SARS-CoV-2. They wanted to rule out that unfavorable Th2 skew.
And how did they measure that? It wasn't just looking at antibodies in the blood, I take it. The sources mentioned some specific techniques.
No, much more detailed than that. They used techniques like ELISpot assays. These showed strong interferon gamma, IFN gamma responses. That's your classic Th1 cytokine signature.
Right.
And importantly, minimal interleukin-4 or IL-4 responses after the second dose. IL-4 is more of a Th2 marker. And then intracellular cytokine staining, or ICS, backed this up. It showed a high frequency of CD4 plus T cells pumping out IFN gamma or IL-2 or TNF, all associated with Th1 responses. But crucially, the frequency of cells making IL-4, very low, negligible really. They even saw circulating S-specific CD8 plus T cells producing IFN gamma.
So a clear Th1 picture across the board in the macaques. And this specific profile had to translate into actual protection, presumably.
It did. That's the key link. This confirmed the desired protective immunological profile was being achieved in a primate model. That gives you high biological relevance for things like neutralizing activity.
Which they measured.
Oh, yes. They measured the VNT50, that's the viral neutralization titer, at 50% against the USAWA 2020 strain, which was, the dominant reference isolate early on.
Right, the original strain.
Confirming neutralization against that strain in a primate gave everyone the necessary confidence that the vaccine was generating the right kind of immune weapons to fight the actual virus out in the world.
Okay, so NHPs are like the dress rehearsal, validating the type of immunity, confirming it works in a system close to ours. But then we often move to rodents for scaling up or different kinds of questions.
Exactly. Rodents, usually mice, are indispensable for testing mechanism, potency, often in more stringent, sometimes lethal challenge models, and you can use more of them, allowing for faster throughput.
Like the MERS-CoV example in the sources, they use some very specific mice there.
Right, the MERS-CoV work. Researchers used these highly specialized transgenic mouse models. They were engineered to express the human DPP4 receptor, HDP4-TG mice, they're called.
Why the human receptor? Why was that necessary?
Because DPP4 is the actual receptor MERS-CoV uses to get into human cells. By putting the human version of that receptor into mice, you create a model that's, well, much more relevant for infection. It makes them susceptible.
Ah, I see. So it makes the challenge realistic.
Extremely realistic, and often lethal in these models. So these HDPP4 mice provide a really stringent test. If your vaccine can protect these mice from a lethal MERS-CoV infection under these conditions, that's powerful evidence. It validates the neutralization capacity in a live in vivo challenge setting. Very hard to replicate that otherwise. Gives you that clear yes-no on protection.
Makes sense. And it's not just for infectious diseases, right? The sources mentioned using mouse models for drug toxicities too, like with CAR T cells.
That's another key application, yes. It shows their utility for predicting specific acute hazards. Take CAR T-cell therapy. A major acute risk is cytokine release syndrome, or CRS. It can be quite dangerous.
That inflammatory storm, right?
Exactly. The sources noted that in humans, this toxic phase often kicks in around 3 to 5 days after the CAR T-cells are infused, sometimes followed by neurotoxicity a bit later, maybe 5 to 7 days.
And mice can actually model that, an acute human inflammatory response.
Surprisingly well, for the acute phase at least. Researchers have developed mouse models specifically designed to mimic the CRS cascade. They replicate that toxic phase.
What's the value of that? Screening potential therapies.
Precisely. It allows for safety screening of different SAR-T constructs or testing interventions to manage CRS. They help researchers dissect the underlying mechanisms, things like vascular endothelial activation, which is a key part of severe CRS. So yeah, rodent models are highly effective for gross efficacy, checking neutralization, and modeling these acute, high-level inflammatory toxicities. They give us those essential go-no-go answers on potency and immediate danger.
Okay, so we've established their value, NHPs for the immune profile and primate-level efficacy, rodents for mechanism, potency, acute tox. But here's the pivot you mentioned earlier. Where did they fall short? What's this critical blind spot regarding long-term human-specific autoimmune risk?
This is exactly where the immunogenetic gap becomes critical. They tell us about the innate immune response, the general T-cell response, they confirm the desired Th1 polarization, which is all vital.
Right.
But they simply cannot tell us what a specific individual human HLA molecule is going to present to T-cells. And since that HLA presentation is the absolute gatekeeper, the prerequisite for activating the T cells that might cause molecular mimicry, well, the models are functionally deaf to that specific personalized human risk. They don't speak the right genetic language, if you will.
All right, let's dive deeper into that hazard itself. Molecular mimicry is a term thrown around a lot, especially in vaccine safety discussions. Can we get a really crisp, robust definition of what it means immunologically?
Okay, so molecular mimicry at its core, it's really a failure of self-tolerance, and it's driven by structural similarity. It happens when an antigen, maybe from a virus, a bacterium, or in this context, a component in a vaccine shares significant sequence homology, or importantly, structural similarity, with a protein that's part of the host, a self-protein.
So the immune system sees the vaccine component or the virus and learns to attack it.
Right. But then, because a self-protein looks very similar, the same immune cells, T cells or B cells, that were trained to fight the foreign invader might then cross-react. They mistakenly attack the host's own tissues because they recognize that similar structure.
It's like friendly fire, almost. Mistaken identity leading to self-attack.
That's a good analogy.
Yeah.
And the consequences can be devastating, leading to autoimmune disease. This mechanism, this cross-reactivity idea, it's the main hypothesis behind many cases of autoimmunity that seem to pop up after viral infections. Think about the suggested links between, say, flu infections and some cases of narcolepsy.
Right.
And to actually confirm mimicry is happening, not just theorize about it. Immunogenicity studies have to get really specific. They use detailed epitope mapping, identifying the exact small fragments of proteins involved, and then do cross-reactivity evaluations. Finding those shared epitopes, those little pieces with similar sequences or shapes, that's the hallmark you look for.
But proving it definitively in humans, especially for rare events after vaccination, that seems incredibly difficult.
Oh, it is. Establishing causality is the major hurdle. But we do have documented examples where molecular mimicry is considered the leading biological explanation for adverse events reported after vaccination. Let's take the measles vaccine and Guillain-Barré syndrome, GBS.
Okay, what's the connection there?
Well, the connection lies in biological plausibility. We know for a fact that GBS can occur as a complication after a natural measles virus infection. So, the theory goes there's a plausible causal link. The antigens in the vaccine, derived from the virus, could potentially trigger the same autoimmune reaction, GBS, in susceptible individuals.
Biologically plausible? Okay.
Yeah.
But is the clinical evidence strong?
That's where it gets murky. There are reports in the medical literature of GBS happening temporarily close to measles vaccination, often case reports, but the problem, frequently, is the lack of detailed clinical or immunological data in those reports. Meaning they might lack specific antibody tests or detailed patient histories, things you'd need to rule out other causes and really pinpoint the vaccine as the trigger in that specific case. So, while the biological plausibility is there, making a definitive causal link in individual reported cases is often precluded by incomplete data. Proving a rare event against the background rate is just really tough.
Okay, so measles GBS is plausible, but hard to prove definitively from case reports. What about hepatitis B vaccine? The sources mentioned Engerix-B in a list of reported adverse events.
Yes, the hepatitis B vaccine, specifically Engerix-B, presents a somewhat different picture, perhaps more suggestive. The manufacturer's own information includes a list of adverse experiences reported during post-approval use, and that list is quite notable.
What's on it?
It includes Guillain-Barré syndrome again, but also things like transverse myelitis, optic neuritis, and even multiple sclerosis or MS-like symptoms.
Those are all neurological conditions, often with an autoimmune component.
Exactly. They're all disorders involving inflammation or autoimmunity in the nervous system. Seeing this cluster of neurological events reported, even if causality isn't proven in every case, has led immunologists and critics to suggest quite strongly that there might be a specific risk related to the viral protein content in the vaccine, the hepatitis B surface antigen, or HBag potentially acting through mimicry pathways targeting neural tissues.
Right, so the pattern itself is suggestive. Now, fast forward to today, and a lot of this searching for mimicry happens computationally first, right, using bioinformatics. What did that approach reveal about SARS-CoV-2?
Oh, the computational effort for SARS-CoV-2 was massive, and the findings were, well, almost overwhelming at first glance. Studies used bioinformatic tools, things like BLAST-VP for sequence similarity, HMR for profile hidden Markov models to compare SARS-CoV-2 proteins against the entire human proteome database.
And what did they find? Just raw numbers?
The raw numbers were staggering. One analysis identified over 150,000 sequence alignments between non-redundant SARS-CoV-2 protein sequences and human proteins.
Wow. 150,000 potential points of similarity. That sounds like a lot of noise. How do you find the real signal, the dangerous mimics in that?
You're right, it's a huge data set. But then you apply filters. You look for specific types of homology, maybe focusing on peptides known to be immunogenic or peptides that match known T-cell or B-cell epitopes.
Okay, so refining the search.
Exactly. By doing that, researchers were able to zero in on potentially cross-reactive epitopes, and they found some specific concerning hits. For example, they identified sequence homology between certain SARS-CoV-2 proteins like ORF6 and the nucleoprotein, N protein, and specific human heat shock proteins.
Heat shock proteins, like HSP90.
Precisely. HSP90 beta, specifically HSP90AB1 and HSP90AA1, showed up. And what's really significant is that the specific shared peptide sequences they found, say between the SARS-CoV-2 N protein and human HSP90 beta, were found to overlap with experimentally validated epitopes already listed in immunological databases like the IEDB.
So it's not just theoretical sequence similarity. The shared pieces are known to be recognized by the immune system.
Correct. It means the molecular blueprint for potential autoimmunity is demonstrably there, embedded in the sequence homology. It suggests a potential trigger exists, waiting perhaps for the right genetic context, the right HLA type in an individual.
Fascinating. And you mentioned this isn't just about vaccines. Pathogens themselves use mimicry.
Absolutely. It's thought to be a pretty common evolutionary strategy for immune evasion. Our sources mentioned HIV as a prime example. HIV is believed to have evolved specifically to protect itself by mimicking key host proteins.
How does mimicking the host protect HIV? Seems counterintuitive.
It creates a dangerous dilemma for the host's immune system. HIV antigens can mimic things like human T-cell receptors, other important host proteins, even proteins from our own gut microbiome.
So if the immune system attacks HIV effectively,
it might simultaneously have to attack the body's own T cells or other vital tissues that share those mimicked structures. This self-inflicted damage, this autoimmunity driven by mimicry, makes it much harder for the host to clear the virus completely without causing significant harm to itself. It's a clever, if destructive, survival tactic for the pathogen.
Okay, we've defined the hazard molecular mimicry. Now let's really zero in on why our standard animal models, which we established are useful for many things, generally fail at predicting this specific risk in humans. You said it comes down to the HLA system.
It really does. This is where we need to get specific,
Yeah.
down to the molecular level. For T cells to kick off that adaptive immune response, the response we want from a vaccine, but also the one we fear in autoimmunity, the CD4 plus helper T cells need to see an antigen.
But they don't see the whole virus or protein.
Right. No, they only recognize short fragments, little peptides. And crucially, these peptides have to be presented, sort of held out for inspection, by special molecules on the surface of antigen-presenting cells, or APCs, think dendritic cells. In humans, these presentation molecules are the MHC class II molecules, which we call human leukocyte antigens, or HLA.
HLA. And the issue is that these HLA molecules are incredibly picky about which peptides they present.
Exquisitely picky. You can think of them like, I don't know, highly customized molecular grooves or pockets. They only bind and present peptides that have the right anchor amino acids at specific positions to fit snugly into those pockets.
That's about the fit.
It's entirely about the fit. We have detailed structural data showing this. Take HLA-DR1, for example. Its binding groove has pockets specifically shaped to accommodate the side chains of amino acids at certain positions within the peptide, usually positions P1, P4, P6, P7, and P9 relative to the peptide's anchor. The chemistry and the physical size of those pockets dictate which peptides can bind. If a peptide doesn't have the right anchors to fit into those specific pockets on that specific HLA molecule, it just won't be presented effectively. The T cell never even gets a chance to see it.
That is incredibly specific. Can you give an example of how even a tiny genetic difference between HLA types changes everything, changes the risk profile?
Oh, absolutely. The HLA-DR4 molecule is a perfect case study.
Yeah.
There are different versions or alleles of HLA-DR4. Let's look at one called DR4-DW4. Structural and binding studies show it has a strong preference for peptides, where the amino acid at that crucial P1 anchor position is aromatic. Think phenylalanine or tyrosine, these bulkier rings.
Okay, so DR40W4 primarily presents peptides with an aromatic anchor at P1.
Correct. Now compare that to two other closely related variants, DR40W10 and DR40W14. They show a completely different preference at P1. They prefer aliphatic amino acids, smaller, chain-like ones like valine or leucine.
So a totally different set of peptides would bind to DW10, DW14 compared to DW4.
Exactly. And this dramatic switch in binding preference comes down to a single amino acid difference in the HLA molecule itself at position beta 86 in the protein chain.
One amino acid change.
One single change. In the variant that prefers aliphatic amino acids, DW10, DW14, there's a glycine at beta 86, which is small.
Right.
In the variant that prefers aromatics, DW4, there's a valine, which is bulkier. That larger valine side chain physically restricts the P1 pocket, making it too small for the bulky aromatic amino acids to fit comfortably. The smaller glycine leaves the pocket open.
Unbelievable. So that single amino acid difference in a person's HLA gene dictates whether a specific peptide, maybe from a vaccine or maybe a self-protein that mimics it, even gets shown to their T cells. That dictates their personal risk.
You've got it. That is the core limitation of the animal models for predicting mimicry. Mice, macaques, they have their own MHC genes, but they don't express the vast polymorphic repertoire of human HLA alleles. There are thousands of different HLA types and combinations in the human population.
So a mouse model, even a sophisticated one, might tell you no problem, but that's because its MHC molecule simply couldn't present the specific human peptide that would cause a problem in a person with, say, HLA-DR4-DW4.
Precisely. The model is genetically blind to that human-specific interaction. You cannot directly predict which human epitopes will be presented and therefore which individuals might be at risk based on their HLA type, using a genetically non-representative animal model.
Okay, so if the animals can't tell us about human HLA presentation, we need to look directly at human systems, right? How do researchers map which epitopes are actually presented by human HLA?
Right, we need human data. The main technique involves sophisticated mass spectrometry. It's called immunopeptidome profiling. For assessing SARS-CoV-2, for instance, researchers took monocytes from human donors carefully chosen to represent different HLA types, specifically HLA-DRB1 alleles in this case.
Okay.
They differentiated these monocytes into dendritic cells, or MDDCs. In the lab, these are key APCs. Then they pulsed these human DCs with the recombinant spike protein from the virus.
Fed them the protein, basically.
Exactly. Then, using mass spec, they could actually identify and sequence the peptides that were being naturally processed and presented by the various HLA-DRB1 molecules on the surface of those human DCs. They captured presentation across 9 different HLA-DRB1 alleles this way.
And what came out of that? How many different spike peptides did they find being presented by just those 9 human HLA types?
The number was quite striking. They identified 209 unique peptide sequences derived from the spike protein that were bound and presented by these human HLA molecules.
209. Just from spike, presented by only 9 HLA alleles. That really highlights the complexity, doesn't it? The sheer number of potential T-cell targets.
It absolutely does. The study also identified hotspots regions within the spike protein sequence that were particularly rich in these presented peptides. For example, the regions around amino acids 2449 and 457-485 were hotspots.
And did this match up with other data? Like, were these regions known to trigger T-cells?
Yes, crucially, there was good concordance between the peptides they identified using mass spec, the immunopeptidome, and T-cell immunogenicity data reported in other studies. It validates this in vitro human system approach. It tells us directly which specific peptide fragments the human immune system can actually see and potentially react to, bypassing the prediction problems of non-human MHC.
Right. It's direct observation versus inference from a different species. Now, just to really hammer home the limitation of models, the sources also mentioned that even within a single animal species, like rats, genetic differences dramatically change the outcome in autoimmunity models.
Yes, this is a really critical point, maybe the final nail in the coffin for relying solely on animal models for mimicry prediction. Look at experimental autoimmune encephalomyelitis, EAE. It's the standard mouse or rat model for multiple sclerosis.
Okay.
Studies compared 2 different common lab rat strains, DA rats and PVG rats. Both can get EAE, but how they get it and how their immune systems respond is fundamentally different based purely on their slightly different genetic backgrounds.
Well, different in what way? Just severity.
Way beyond just severity. We're talking deep mechanistic differences at the gene expression level. When they profiled the T cells involved in the autoimmune attack, PVG rat cells turned up a whole set of potassium channel genes that weren't significantly changed in the DA rats.
Okay, different genes activated.
And when they restimulated the T cells with the autoantigen, the self-protein that triggers EAE, the difference was huge. In DA rats, about 903 genes showed differential expression. In PVG rats, 1,356 genes.
Wow. Over 450 more genes behaving differently just because of the rat strain.
Exactly. Same disease model, same autoantigen, but fundamentally different molecular pathways being engaged, affecting T-cell activation, cell death pathways, apoptosis, transport mechanisms, you name it.
So the takeaway is...
The takeaway is that even these minor controlled immunogenetic differences between 2 rat strains completely alter the underlying autoimmune response. If we see such dramatic variation between rat strains, how can we possibly expect any single rodent finding, especially a no-problem found result, to reliably predict the risk across the vast genetic diversity of the human population with its thousands of HLA combinations? It underscores the challenge of translation. It's simply not a safe assumption for this kind of personalized HLA-driven risk.
Okay, so it's not just which peptide gets presented by HLA, but also what happens next, right? The type of T-cell response that gets triggered is crucial in deciding between protective immunity and autoimmunity. This involves different T-cell subsets, especially this balance between Th17 and Treg cells.
Absolutely. That differentiation step is critical for maintaining what we call peripheral tolerance, basically, keeping the immune system in check outside the initial development stages. When naive T cells encounter an antigen presented by HLA, they get signals from their environment, mostly cytokines, that tell them what kind of T cell to become.
And sometimes are good, and some are bad in the context of autoimmunity.
Well, good for tolerance are the regulatory T cells, Tregs. They are absolutely essential for preventing autoimmune reactions. Their development and function are typically driven by the transcription factor, FOXP3. Their job is to suppress inappropriate immune responses.
And high levels of certain inflammatory signals can shut them down.
Yes, for instance, high concentrations of IL-6 combined with TGF-beta can often inhibit Treg function or development, shifting the balance away from tolerance.
Okay, so Tregs are the brakes. What about the accelerator for autoimmunity?
That would often be the T helper 17 cells, or Th17 cells. These are strongly implicated as key drivers in many autoimmune diseases. Their differentiation is critically dependent on pro-inflammatory cytokines, particularly IL-6 and IL-23.
IL-23 shares a subunit with IL-12, right? The P40 is subunit.
That's right, the P40 subunit is shared. And these Th17 cells are characterized by producing their own set of inflammatory cytokines like IL-17A, IL-17F, IL-22. Sometimes they even co-produce IFN gamma, blurring the lines a bit with Th1 cells. But it's this Th17 profile that's essential for driving disease in many animal models, like the EAE model we discussed, and also models like experimental autoimmune myocarditis. EAM.
But here again, the models get complicated, don't they? The sources mentioned conflicting results about TGF-beta's role in EAE, sometimes needed for TREGs, sometimes maybe for Th17.
This is a perfect example of the complexity and why translating model mechanisms can be tricky. TGF-beta is classically thought of as promoting TREG development, which would be protective against EAE.
Right.
However, in some models, the differentiation of the pathogenic Th17 cells themselves actually seems to require TGF-beta acting together with IL-6. It primes them in a way.
Okay, so it can play both sides.
It seems so, and to make it even more confusing, in one specific mouse model, researchers genetically deleted the receptor for TGF-beta on T cells. And you might expect, if TGF-beta helps drive pathogenic Th17, that this might make EAE worse or have no effect. But instead, it completely prevented EAE.
Wait, removing the TGF-beta signal prevented the disease, even though TGF-beta might help make the bad Th17 cells. How does that work?
It suggests a really nuanced role. Perhaps TGF-beta signaling is required at some very early stage, maybe for T-cell survival or activation in general, even if its continued presence later helps push toward that Th17 phenotype. Or maybe its effects are different in different cellular compartments or at different times. The point is, the role isn't simple or linear.
It really highlights how, even if an animal model gets the disease, the precise molecular levers being pulled might be subtly different from humans, or even from other models.
Exactly. The pathway complexity and its sensitivity to the specific genetic background and environment make direct translation of these mechanistic findings a major interpretive leap, especially when we're talking about safety predictions.
Okay, given these T-cell modeling complexities, let's circle back to mimicry itself. There was a study mentioned that looked directly at human B cells, trying to get functional proof of cross-reactivity in a human cohort, focusing on that hepatitis B vaccine antigen again.
Yes, that was a fascinating study. They took B cells from human subjects and tested their reactivity to peptides from the hepatitis B surface antigen, SHBSA. But they also tested reactivity to peptides designed to mimic parts of myelin oligodendrocyte glycoprotein, or MO.
MO, that's a protein in the nervous system, right? A target in MS.
Exactly. MOGO is a known autoantigen in multiple sclerosis and related demyelinating diseases of the central nervous system. So comparing reactivity between the vaccine antigen peptides and the MOG mimics gets right at the heart of potential cross-reactivity relevant to neurological autoimmunity.
And what did they find in these human B cells? Was there evidence of cross-reactivity?
The findings were quite striking. They found that reactivity to at least one of the MOG mimicking peptides was present in B cells from 53% of the human cases they studied.
Over half the people had B cells that could recognize a MOG mimic.
Yes. And perhaps even more telling, they looked for double reactivity, meaning B cells that reacted to both an SHB ag peptide and its corresponding MOG mimicking pair. That double reactivity was also found in 53% of the cases.
53% again, that seems high. It suggests that a significant portion of the population might already have B cells capable of recognizing structures shared between the Hep B vaccine antigen and a self-protein like MELO.
That's the profound implication, yes. It suggests these cross-reactive B-cell clones might be part of the normal pre-existing immune repertoire in many individuals, potentially just waiting for the right trigger like vaccination or infection to become activated.
Did they identify which specific parts of the Hep B antigen were involved?
They did drill down into the specifics. Overall, 92% of the cases showed B-cell reactivity to some SHBS peptide. But critically, all of those reacting cases showed reactivity specifically against one peptide region, SHBSH 130-149.
So very specific targeting within the vaccine antigen in the human response.
Extremely specific. And that level of detail identifying the precise peptide sequence involved in cross-reactivity within a human cohort is exactly the kind of information you simply cannot reliably get from a standard rodent or even NHP model.
Because their MHC-HLA is different, and their B-cell repertoire might be shaped differently too.
Precisely. So while animal models can be useful for modeling the general bias of the T cell response, Th1 versus Th17 as we discussed, they fundamentally cannot replicate these specific peptide-driven cross-reactivity profiles seen in human cohorts. Those profiles are dictated by the unique landscape of human HLA expression and the resulting T and B cell activation patterns. This human-specific functional data really is the gold standard that highlights the limitations of animal models for predicting this particular personalized risk.
So, summing up this section, models can show us general pathways like Th17 TREG balance, but the actual trigger, the specific cross-reactive epitope recognized in the context of human HLA that requires looking directly at human systems or predictions based on human genetics. Hashtag tag outro. Okay, let's try and synthesize all of this. We've covered a lot of ground. We've clearly seen that non-human primate models are invaluable, right? They give us crucial data on vaccine efficacy, on T-cell polarization, confirming that protective Th1 bias we want.
Absolutely. Essential data points, yes.
And rodent models, they help us dissect pathogenesis mechanisms, acute toxicity like CRS, things that need higher throughput or lethal challenge models.
Definitely. They answer important questions about potency and immediate dangers.
But the Achilles heel, when it comes to predicting that rare but potentially devastating event of molecular mimicry triggering autoimmunity, seems to be the uniquely human immunogenetic architecture, specifically the incredibly polymorphic HLA system.
That's the core message. We really have to move past thinking any single animal model, no matter how sophisticated, can adequately represent the sheer diversity of human immunogenetics relevant to T-cell recognition.
The numbers you mentioned were staggering. 209 unique spike peptides, presented by just 9 human HLA types in that one study.
Exactly. And the MOG-SHB-AG B-cell study showing 53% double reactivity in a human cohort. These results force us to confront the reality. Epitope presentation and potential cross-reactivity in humans is incredibly complex and personalized. That complexity just can't be captured reliably in a cage full of genetically identical mice or even a small group of macaques.
So this really mandates a shift, doesn't it, in how we approach safety assessment for this specific risk. We can't just rely on the animal models to predict human mimicry events that are defined by human HLA.
I think it absolutely does. We have to lean much more heavily on immunoinformatics up front, using tools like BLAST-TP and HMRA for sequence homology searches, yes, but crucially, tools like NetMHC-IPAN that specifically predict peptide binding affinity to a wide range of human HLA alleles.
So computational screening based on human HLA data.
Yes, combined with functional human in vitro systems like the dendritic cell profiling we talked about, the explicit goal has to be proactively identifying potential cross-reactive epitopes, danger signals, using these computational and human-based methods before a vaccine candidate even gets near large-scale human trials.
It's about flagging the risk based on the human genetic blueprint itself.
Precisely. Given the stakes involved with autoimmunity, this kind of comprehensive, in silico and in vitro preclinical screening focused on human HLA interactions is rapidly becoming an essential first line of defense for personalized vaccine safety. It's about predicting based on the relevant genetics.
That transition, moving some of that predictive burden from the wet lab animal models to computational algorithms trained on human data, it makes a lot of sense when the critical variable is human genetics.
Yeah.
It fundamentally changes the approach to assessing this particular risk.
It does. And that leads us directly to our provocative thought for you, the listener, to mull over.
Okay, let's hear it.
Given the ever-increasing power of bioinformatics and AI to identify these subtle sequence and structural conservations between pathogen proteins and self-proteins, specifically in the context of predicted HLA binding, will the future of vaccine safety assessment for mimicry risk essentially abandon traditional animal models for this specific purpose?
Wow, abandon them entirely for mimicry prediction.
Well, relying instead almost wholly on high throughput in silico HLA binding predictions and functional human cell assays to screen out potential autoimmune triggers before they ever reach animal testing, let alone human trials. Think about it. Are we moving towards modeling human genetic diversity relevant to autoimmunity primarily through computation rather than through expensive, time-consuming, and potentially genetically misleading in vivo animal studies?
That's a huge question. Trading the animal model for the algorithm when it comes to predicting these personalized HLA-driven risks. What does that mean ethically, practically, regulatorily? Definitely something to think about. That puts us right on the cutting edge. Thank you for walking us through this incredibly detailed and complex topic. And thanks to our listener for the sources. We really appreciate you joining us for this deep dive. We'll see you next time.